Hello, genetics friends. Let's talk about Mendelian ratios and exceptions to genetic Mendelian ratios. Genes are present on two homologs, like two chromosomes, right? One from your mom, one from your dad. They're segregated and assorted independently during gamete formation, so you don't actually know which one you're going to get from which parent. Ratios are modified when gene expression does not adhere to one of these rules. Have simple dominant or recessive, right? So that's a blood type. That's a pink in the middle of red and white, where there are more than one pair of genes that influence that trait. Otherwise, your classic ratios are 3:1 or 9:3 to 3:1. So in diploid organisms, that's you're a diploid organism, with a homologous pair of chromosomes, also you, two copies of each gene, also you, influence the inheritance of traits. Each copy of the gene is called an allele, and alleles occur within populations, and that's how we do population genetics. Genes on the X and Y chromosome. So some modified genetic Mendelian ratios are when you get a sex-limited or sex-influenced inheritance. So this is when all of a sudden you have to do your ratios and you have to include the X and Y chromosome. And some of these are sex-influenced because women can only have characteristics related to like uteruses or things like that only women have or only men have. So they are influenced because, right, men don't have a uterus, so it doesn't affect them. Expression of the traits can vary depending on the overall environment in which the gene is exposed. So we'll talk about Himalayan rabbits and things like that. Again, we have alleles. Alleles are the alternate forms of the same gene. So big A, little a, whatever you want to call it. They are the wild type is normal, and we only say that in terms of like flies or things that have diseases. We don't say brown hair is normal and everything else is not normal. The wild type is the allele that occurs most frequently in the population. Again, this is going to be including flies and some other animals in which there is a normal type that we would say, this is the normal color of mice, this is the normal color of flies, things like that. Mutations are the source of modified genetic information. So the mutant allele contains modified genetic information and often specifies an alternate gene product. So red eyes are normal in flies. White eye is a mutation, and it is a recessive mutation, so it is a modified genetic information. Okay, so some new phenotypes, remember phenotype is what it looks like, results from a change in function. So this can be a loss of function mutation, so it doesn't function at all. This would be albino, most diseases, or a gain of function mutation. This would be it gains a more vibrant color or a darker color or a neutral mutation. When we get into molecular, we will talk about these kinds of mutations. Note that phenotypic traits may be affected by a single mutation in just one gene. Again, we will talk about that when we get to sickle cell anemia and some other things. So a loss of function mutation. Mutation in the gene causes change in the enzyme, the conformation of the enzyme, the folding of the enzyme. If the loss is complete, it's a null allele. So it just doesn't have any effect. It nulls out the whole thing. A gain of function is an enhanced function of a wild type. This increases the quality of gene products by affecting regulation. And neutral, the gene product presents no change to the phenotype. So there might be a change in the DNA or the genotype, but there's no phenotypic difference. Symbolizing alleles, typically tall is dominant. The dominant one is a capital letter. The recessive one is a lowercase letter. Again, Drosophila and fly mutation are a little bit different. And then when we do things that are codominant, so when you think of things like blood type, we're going to do AO, AB, OO. So sometimes that looks a little different. But typically, uppercase refers to dominance. And then, of course, Drosophila mutation is a little bit different. Ebony mutant is E. Normal wild type is E+. So this is the normal wild type of the fly. Then you get a heterozygous. Phenotypically, still wild type. Genotypically, this one is both a mutant and phenotypically it has the ebony phenotype. You can do it without the E's and just put the pluses in the E's. You'll see that both ways. Variation is used when no dominance exists between the alleles. So this is AB. A is not dominant to B. They just both express. You have both the antibodies for A and B. This would be an uppercase italic letters and superscripts used to denote alternative alleles. Functions of genes or disorder caused by a mutant gene. There are a lot of ways to write these down. Typically, you just have to look them up depending on what organism you're looking at. If you're looking at C. elegans or plants or yeast or bacteria or humans. Yeast CDK represents the cyclin-dependent kinase gene. It's a product involved in cell cycle regulation, and it's often experimented with. Bacteria, the LEU or LU refers to the mutation that interrupts the biosynthesis of amino acid leucine. The symbol DNAi represents bacterial genes involved in DNA synthesis, and some of us have heard of the BRCA1 gene, and it's associated with breast cancer. So you can see there's just a lot of ways that we make a symbol for these genes. There's not one, not everything's going to be big A, little a. We have talked about this in class. We've done some problems like this where we have the intermediate phenotype results from a cross between parents with contrasting traits. So this is our A, big A, little a, but in the middle you get pink, right? So you have a dominant red, white is recessive, but in the middle, this is just an incomplete. There's not enough red pigmentation to make red, so you get pink. It's just a watered down pink, if you will. This happens in snapdragons. They have incomplete dominance. So your F1, let me review this. F1 is the filial generation. So that is the parents. And so the F1, the parents, the offspring of the parents have pink flowers. And what you did here probably was did a red with a white, and you got the F1, which is pink flowers. Now you're going to breed the F1s together. I know they're brothers and sisters, but it's plants, so it's totally fine. And you're going to get a 1 to 2 to 1 ratio, right? A fourth red, a half pink, a fourth white. In this case, each genotype has its own phenotype. Does that make sense? Because the heterozygous have a different phenotype. Incomplete dominance. There it is. Again, we've talked about it. Tay-Sachs disease, it's a biochemical disorder. The homozygous recessives affected by a fatal lipid storage. So the hemosaccharomyces A activity is absent, and the enzyme involved in lipid metabolism. Normal heterozygous has one copy of the mutant gene. Half of the enzyme activity compared to the homozygous normal carriers. So you're normal, you have some effects, or you completely have the disease. So this is saying that the heterozygous individuals carry one copy of the mutant gene. And they have half the enzyme activity. Codominance. Let's see if we can get this one in really quick before this video ends. This is what we're saying. 2 alleles each produce a product. So one produces black, one produces white. Together, you have black and white fur that looks brown. Joint expression. Both are producing their A, B, codominance. One produces A, one produces B. They're codominant. A isn't dominant or recessive over B. There's not really a dominant or recessive. A isn't dominant over recessive. They're using the MN blood group. I use the AB. This is just a different blood group in humans. It's characterized by the antigen glycoprotein. 2 forms of the glycoprotein exist, designated MNN. Another type of blood type. Individuals may exhibit either one or both. You're either M, you're N, or you're MN. And this is under the control of chromosome 4. So here's the MN blood groups. You're either M, you're MN, or you're N. There's no O. It makes it a little bit easier. Mating's